Morning, everyone. We're going to give it a couple minutes. Good morning. We're going to give it one more minute and then we will start. Okay, I will get started. Good morning, everyone. So, welcome. We're so glad you could join us for today's NIH Office of Dietary Supplements seminar on mechanisms. I'm Jasmine Hayes, and I'm a health scientist administrator with ODS. In addition to organizing our seminars, I serve as the lead for our co-funding program. Before we welcome today's speakers, let me take a moment to go over a few brief announcements. I'd like to remind you that our next ODS seminar is coming up soon. This seminar will discuss immunity and resilience and will be held on March 25th. If you're not already on the seminar's mailing list and would like to be, please check the chat. We'll share an email address there, and you can use it to request being added to the mailing list. Now, next, a quick note about webinar logistics. To ensure good audio quality, everyone has been muted. During the Q&A, you can raise your hand, and we will unmute you so you can ask your questions. If you experience any technical issues with audio, video, or closed captioning, please use the chat and one of us will be happy to assist. And so as a reminder, please be considerate that everyone can fully enjoy and learn from our today's speakers. So now I have the pleasure of introducing our first speaker, Dr. James Collins, or as he goes by Jamie. He is a professor of human nutrition in the Food Science and Human Nutrition Department at the University of Florida. He has broad training and expertise in physiology, molecular biology, genetics, nutrition, and has been involved in biomedical research for more than 25 years. His research research interests include molecular, I'm losing my voice. Sorry, I'm coming down with something. His research interests include molecular mechanisms of intestinal iron transport, the influence of copper on iron homeostasis, nutrient gene interactions, and iron absorption during inflammatory states. Ongoing studies in his lab relate to iron supplementation and how this negatively impacts copper homeostasis, as well as development of therapeutic approaches to modulate intestinal iron transport in various disease states. He is associate editor of the Journal of Nutrition. Previous positions include graduate coordinator and director of the Nutritional Sciences doctoral program in the Food Science and Human Nutrition Department at the University of Florida, assistant professor at the University of Buffalo, and a research associate professor at the University of Arizona. He received his master's in molecular biology from Middle Tennessee State University, his PhD in molecular physiology from Vanderbilt University, and completed a postdoc in transport physiology at the University of Arizona. So now I will give it over to Jamie to give his presentation.
Excellent. Can everybody see this?
Yes, we can see it.
Excellent. All right. Well, Jasmine, thank you very much for the nice introduction. It's my pleasure to be here to talk about some of our research that's been going on for the past 20 or more years. I've modified the title a little bit, and here I'm calling this talk Therapeutic Strategies to Modulate Iron Absorption in Human Disorders. So my objectives today are first to outline the basics of iron homeostasis. My main focus will be on intestinal iron absorption. Going to think about heme iron versus non-heme iron absorption. Discuss iron-related disorders in humans, including iron deficiency anemia, hereditary hemochromatosis, and beta thalassemia intermedia. These are iron-loading disorders, these latter two here. And I'd like to also list novel approaches to modulate iron absorption. For example, enhancing iron absorption in iron deficiency anemia and blocking iron absorption in the iron overload disorders, hereditary hemochromatosis and beta thalassemia intermedia. So first, a little background about systemic iron homeostasis. Dietary iron doesn't meet physiologic needs. That iron is recycled. That provides most of the iron. Senescent red blood cells, which contain a lot of heme and most of the iron in the body, undergo erythrophagocytosis by reticular endothelial macrophages. Within those macrophages, the hemoglobin is degraded, the iron is released from the hemoglobin. It can be stored in the macrophages or it can be released to be picked up by transferrin in the blood. Most of the iron in our body is destined for the bone marrow to support erythropoiesis. Importantly, there's no active regulated excretory mechanism for iron. There are daily losses, but these are minimal, about 1 to 2 milligrams per day, and those are non-specific and unregulated losses. So what that means for iron is that intestinal iron absorption is the homeostatic checkpoint that controls overall body iron levels, and this is a unique aspect of iron metabolism. Also, excess iron deposited in tissues is toxic. It creates oxidative stress. So here are a list of pathological outcomes associated with iron deficiency anemia. This is probably familiar to most of you, so I really won't spend any time on it. In iron overload, such as hemochromatosis and beta thalassemia, iron accumulates in various tissues like the liver, the pancreas, gonads, joints, and heart. And you can see some of the pathological outcomes there. Arthropathies, joint pain, diabetes, impotence, etc. A little bit about molecular regulation of iron homeostasis. The main player is hepcidin. It's a peptide hormone produced by the liver. This is thought to be the iron regulatory hormone. This is kind of what it looks like. It's got multiple disulfide bonds, very complex three-dimensional structure. It functions by binding to the iron exporter ferroportin and causes it to be internalized and degraded. A little bit more about that later. And here's actually a diagram of the ferroportin transporter with hepcidin lodged in there, and it is triggering internalization and degradation of ferroportin, and it can also block the iron pathway through there. So collectively, hepcidin functions to block iron release from cells that express ferroportin, and those include duodenal enterocytes, the reticular endothelial macrophages, and hepatocytes. When elevated iron stores occur, when there's inflammation, that increases production of hepcidin, while conversely, iron deficiency, ID, and hypoxia and increased erythroid demand all decrease hepcidin production. In mice, if you knock it out, that causes iron overload. If you overexpress it, that causes iron deficiency anemia. And so here's a simplistic version of how hepcidin functions. I like this particular diagram here. It's simple. This is from Ellen Namath and Tom Gans, a paper they published some time back, and it shows ferroportin as a spigot. And so when hepcidin is around, it is closing this spigot. It's blocking iron absorption, iron release from the spleen and from the liver. Here's a more complex version of this that I'll spend a little bit of time on. This is from a review article published in Cell back in 2004. I've adopted it for my own needs here, but I'll walk you through this sort of one step at a time. I know it's kind of complex, but we've got dietary iron. That would be heme iron derived from animal sources, and non-heme iron we consume about 18 milligrams per day. Of that, about 1 to 2 milligrams is absorbed. That is picked up by transferrin in the blood, and that distributes iron to tissues of the body. Most of that transferrin iron is bound for the bone marrow to support the production of billions and trillions of red cells every day. Those red cells have a finite lifetime. When they become senescent or they're dying, they are engulfed by reticular endothelial macrophages. That's called erythrophagocytosis. Those macrophages can store the iron, or they can later release it, and it goes back into the blood to be picked up by transferrin. And so here is hepcidin made by the liver. When it is produced, it blocks iron release from ferroportin, whether it be in the liver, the reticular endothelial macrophages, or in the duodenum. The gene that encodes hepcidin, called the HAMP gene, is highly regulated. Transcription of that gene goes up when body iron stores are elevated during inflammation, and this process here is mediated by pro-inflammatory cytokines like IL-6. Conversely, HAMP expression and hepcidin production is suppressed when iron stores are low, when there's hypoxia, or when there's increased demand for iron. And so any tissue that expresses ferroportin is potentially a hepcidin target. So I'm going to spend some time talking about this process here of intestinal iron transport. This is it's a diagram from a review article published about 12 years ago. It shows the various sources of dietary iron at the top. We have heme. We don't know exactly how heme is absorbed or how heme is processed within cells. More on that later. We've got a ferrireductase, which reduces non-heme iron from the ferric to the ferrous state. Iron can then be transported into cells by divalent metal ion transporter 1, DMT1. Within the cell, iron can be stored in ferritin, or it can be targeted to the mitochondria, where it supports oxidative phosphorylation. Intracellular iron chaperones have been identified in the last few years that probably mediate iron trafficking through the cell. After the iron enters the cytosol, if it's not stored or utilized, it could be transported out of the cell by an exporter called ferroportin, which is actually the hepcidin target. And then iron is oxidized by a peroxidase called hephaestin back to the ferrous state, where it can then bind to transferrin. And so our interest has been on DMT1. And I'll spend a little bit more time talking about this particular transporter here. DMT1 is a ferrous ion proton cotransporter. It was originally discovered when the mutant gene in the Belgrade rat was mapped out. It's the same mutation, same mutant gene in the MK mouse. So a lot of what we know about DMT1 came from these animal models. It turns out that DMT1 has dual functions. One is in intestinal iron absorption, that's non-heme iron absorption, and this is a blow up of that diagram I showed you on the previous slide. Secondly, DMT1 is involved in cellular iron acquisition via the transferrin cycle. DMT1 activity expression goes up very strongly in iron depletion and hypoxia. Mutations in DMT1 cause severe iron deficiency anemia in humans and in rodents, so there is a clear human correlate to rodent work with DMT1. Intestine-specific DMT1 knockout mice also have very severe iron deficiency anemia, and this is when they're fed diets containing standard rodent diets containing only non-heme iron. Importantly, though, DMT1 may be a target of hepcidin released within the lumen of the gut. This has been something that's recently published in Blood. Quite exciting paradigm there. And also DMT1's role in intestinal heme iron as absorption is unknown, and that is something that we have been testing in the lab recently. So let's start with iron deficiency anemia. Iron deficiency occurs over sort of a series of pathological events. So the first stage of iron deficiency is where there's a decrease in iron stores in reticular endothelial macrophages and hepatocytes. Eventually, there's a defect in the transport iron that is being delivered from stores to the bone marrow. Eventually, that leads to iron-restricted erythropoiesis, where you get decreased hemoglobin synthesis. And then the most severe manifestation of iron deficiency, of course, is anemia, and that's a hypochromic microcytic anemia. Iron deficiency anemia is a leading risk factor for disability and death worldwide. Billions of people likely affected. The World Health Organization estimates that in developing countries, 39 to 48% of children, 42% of women, up to 60% of pregnant women are all iron deficient. You can kind of see that mapped out here on this flat version of the globe with the countries with the most severe iron deficiency in these darker colors. Excuse me. The most common treatment for iron deficiency anemia globally is oral iron supplementation. Most commonly, iron salts, which are a form of non-heme iron, are utilized, such as ferrous sulfate. The problem with these is bioavailability. Treatment with iron salts can induce hepcidin, which can block absorption of subsequent supplemental iron doses. And due to this low bioavailability and poor absorption, supraphysiological doses are often required. And what that means is a lot of excess iron is going to remain in the lumen of the gut, and it can have a lot of GI side effects. It can irritate the mucosa, cause mucosal damage, it can alter the gut microbiome in negative ways, etc. And so ongoing research is trying to identify better oral iron supplements that have higher bioavailability, lower GI side effects, and that are not regulated by hepcidin. And this is a diagram showing many of these different novel forms of iron supplements that have been developed in recent years. This left side here shows the typical ferrous salts that are used. Here's something called iron polysaccharide complex. A newer supplement is heme iron polypeptide. We can have iron amino acids complex and chelates, and there's also things like nanoparticle iron. And these different forms of iron are utilized in different ways. They're absorbed in different ways. Some of these can bypass the normal regulatory routes that have evolved in humans and mammals for iron absorption. We have done some work specifically on iron and amino acids, and I'll share a little bit of that with you here. So this is a paper we published in Blood Advances in 2022. You can see the title here: 4 amino acids increase DMT1 abundance and enhance iron absorption. The key points are outlined here. I'm just going to show you a little bit of data from this paper. So this diagram, or this figure here, is showing that this 4 amino acid formulation stimulated iron 59 absorption in iron deficient mice, and you can see that right here. Without the 4 amino acids present, the iron absorption was about 4% of the dose, and that went up significantly in the presence of the amino acids. Other parameters went up as well. They just didn't quite reach statistical significance. And that same phenomenon was retained in the presence of 1.75 millimolar iron sulfate, suggesting that the 4 amino acids can enhance iron absorption in the presence of a dose of supplemental iron. So we concluded that select amino acids enhance iron absorption by inducing DMT1 trafficking onto the apical membrane of enterocytes, and we speculate that further refinement of this approach could allow iron repletion at lower effective doses. And this is just a summary. This is our graphical abstract from that paper. So let's turn our attention now to iron overload, genetic iron loading disorders such as hereditary hemochromatosis. There's multiple mutant genes that can cause hemochromatosis. What they have in common is the protein products of these genes are all required for hepcidin production. So in hereditary hemochromatosis, we have inappropriately low hepcidin, which leads to high iron absorption and iron accumulation, iron loading in various tissues. A similar thing occurs in beta thalassemia intermedia. This is a condition where we have mutations in the HBB gene, which encodes the hemoglobin subunit beta. Beta thalassemia intermedia is iron loading anemia. This is a form of thalassemia that is non-transfusion dependent. And because of the high erythroid demand in beta thalassemia intermedia, hepcidin again is inappropriately low. You have high iron absorption, and that equates to iron loading. So the mechanisms of iron loading in hemochromatosis include elevated iron absorption from low hepcidin. Plasma iron exceeds the transferrin binding capacity. You end up with non-transferrin bound iron, which is pathologically taken up into cells. Iron accumulates in parenchymal tissues. Free iron is toxic, reactive. It creates oxidative stress, tissue damage, and eventual organ dysfunction. So the treatment for these disorders is typically phlebotomy or iron chelators. So phlebotomy is, of course, not specific for iron. It may have to occur weekly. Maintenance phlebotomy may be needed over a lifetime. And the iron chelators, which are used in thalassemia and beta thalassemia intermedia, they have numerous side effects. So new approaches are being developed. And one thing that we and others have thought about is knockdown or blocking DMT1. The rationale is that we know DMT1 is required for heme iron absorption. DMT1 may be a hepcidin target, as we've learned in recent months, but we still have a key question. Could DMT1 also be involved in heme iron absorption? And that is something that we are considering. So a little bit of background. These are published studies targeting intestinal DMT1 in hereditary hemochromatosis and beta thalassemia intermedia. This paper published in Molecular Therapy in 2019, we were able to show that if we used nanoparticle-mediated DMT1 siRNA delivery via the oral route, we could prevent iron loading in a mouse model of hemochromatosis. We went on and published a paper in Nutrients that showed that we could reverse iron loading once it had already set in, using, again, a hepcidin HAMP knockout mouse model. This is our graphic abstract from that work shown right here. And then we also did a similar study in beta thalassemia intermedia mice, and we showed the same thing. So what do we show from these studies? DMT1 is required for iron loading in preclinical mouse models of these human diseases. Targeting intestinal DMT1 could be an effective adjunctive treatment for iron overload. So we went on and developed new models of heme iron absorption. These have been published recently. Our intent is to then try to discover some new mechanisms of heme iron absorption. So we have, here's the experimental design here. We fed rats and mice either diets that contained iron from porcine red blood cells. Those contained 85% heme iron and 15% non-heme iron. And then we used non-heme iron diets, which are ferrous sulfate. And we tested the ability of these diets to, one, resolve iron deficiency anemia, two, to support normal pregnancy outcomes, and three, to precipitate iron loading and HAMP knockouts. I'm not going to show you the data from the pregnancy outcomes, but both of these diets, the heme iron enriched diet and the ferrous sulfate diet, both promoted normal pregnancy outcomes. I'll show you a little bit of data on iron deficiency anemia. This was a iron depletion repletion experiment where we depleted mice of iron for 3 weeks and periodically bled them to check their hemoglobin. We got their hemoglobins down to 5, and then we repleted them with a variety of diets. And you can see this blue line here was a control 50 ppm non-heme iron diet. A 60 ppm heme iron diet repleted the mice almost exactly the same rate, whereas if we used lower amounts of iron, whether heme iron or non-heme iron, they were not fully repleted. And then as far as the iron loading experiments go, we were able to show that heme iron does contribute to iron loading in hepcidin knockout mice. And you can see that here. They have elevated transferrin saturation. Non-heme iron is blue, heme iron diet is red. And you can see that serum iron is elevated, and they have significant increases in liver iron as well. So elucidating mechanisms of heme iron absorption could inform the development of new approaches to modulate iron absorption in disease states. We went on and did some studies also, and these are unpublished studies from our lab using dietary heme iron polypeptide, which is a new iron supplement. This is again a depletion repletion experiment. We used diets that contain porcine red blood cells, heme iron enriched, and also heme iron polypeptide diets. We compared the two, and you can see that both of those diets repleted these iron deficient rats at the same rate, very similar to the rate that the control non-heme iron diet did. They ended up sort of at the same place. You can see that it didn't matter what diet the animals were on, the body weights were similar. Hematocrit was corrected. Serum iron levels were restored, and TSAT was corrected as well. So here we developed a nutritional paradigm to study heme iron absorption. Next, we wanted to use these models to test the role of key transporters or enzymes in heme iron absorption. And so iron derived from heme is highly bioavailable. Absorption increases during iron deficiency. We know that heme iron contributes to iron loading in hemochromatosis, but the mechanisms of heme iron absorption remain mainly unknown. Best case scenario is that we have receptor-mediated endocytosis. Heme is transported out of the endosomes into the cytosol, where it's catabolized by heme oxygenase in the ER. It's released and joins the intracellular iron pool. We're considering an alternative pathway that's been proposed by others, where heme is catabolized in endosomes and lysosomes and then released into the cytosol by DMT1. That is shown in this diagram here. I unfortunately don't have time to go through this, but the idea is heme comes in, can either be pumped out and be degraded in the cytosol, or it could be catabolized in the endosome. Then you would need DMT1 to pump it out into the cytosol. So we did an iron physiology study first. We put mice on different diets. We weaned them on the different diets, either heme iron or non-heme iron diets, and these were control and inducible DMT1 intestine-specific knockout mice. What we showed is that the control 50 ppm non-heme iron diet and the control red blood cell diet, they both allowed the normal age-related increase in hemoglobin levels from 3 to about 6 weeks of age. However, the DMT1 knockouts didn't matter if you give them heme iron or non-heme iron. They could not utilize that to support the increase in hemoglobin during aging or during postnatal development. You can see that at the end of the experiment, both of the DMT1 intestine-specific knockout groups were anemic. They had very low serum iron. HAMP expression was essentially shut off, encoding hepcidin, and you see that there was an induction of EPO, suggesting increased erythroid demand. We then went on and did an iron absorption study, and we injected donor rats, iron-deficient donor rats, with iron 59. We collected the blood, and we utilized that blood to gavage experimental mice. And what we were able to show is we have 3 different groups here just to set the stage. We had sort of a normal heterozygous control animal. We had an iron-deficient heterozygote, and we had iron-deficient DMT1 specific knockout. And you can see that both of this is a control group. Both experimental groups were highly anemic. And interestingly, what we saw is the heterozygote, which is phenotypically normal, they drastically increased heme iron absorption, but we've completely lost that effect in the iron deficient knockout mice. You can see similar trends in blood, spleen, bone marrow, liver, heart, kidney. So intestinal DMT1 is required for heme iron absorption during iron deficiency anemia. We also have complementary experiments we did in the Belgrade rat, which I don't have time to show you. So overall, DMT1 is required for the absorption of both main forms of dietary iron, Enhancing DMT1 expression or activity could improve absorption of oral iron salts and heme iron-based dietary supplements during iron deficiency anemia. And blocking intestinal DMT1 could be effective adjunctive treatment to mitigate iron loading in conditions such as hereditary hemochromatosis and beta thalassemia intermedia. So here's our funding from NIDDK, the Office of Dietary Supplements, past and current grants, and a bunch of the people involved in these studies. And these are all University of Florida nutritional, former or current nutritional sciences doctoral students. All right, thank you very much. Happy to take any questions.
Thank you very much. We have time for, one question. We can also, if we have some time at the end, we can save time for questions. So we have a question of how consistently have rodent models of iron absorption or metabolism translated to clinical results? And when amino acids are added to oral iron and absorption is improved, are there fewer GI AEs?
Those are great questions, and these are actually things that I think about all the time. I don't often see people justify using rodents for doing a lot of things, but I think that mouse research in particular has been very informative in the iron field. Iron metabolism in mice and rats is very similar to humans, so I think they've been very useful. DMT1 plays a similar role in both species. Hepcidin does. So I think those things do translate very well. As far as amino acid work goes, what was the specific question again there? Could you run that by me again about the amino acids?
When amino acids are added to oral iron and absorption is approved, are the fewer GI AEs? And then also the follow-up was, are those free added amino acids or they're in peptides?
So the work we did was just adding those amino acids. We actually tested all 20 amino acids, and we found 4 that tended to enhance iron absorption or enhance DMT1 expression, so we combined them together. I think there is some suggestion that chelation of iron with amino acids or peptides does decrease GI side effects. I don't think we know exactly how they're absorbed. But the idea would be is if you could use amino acids or peptides to enhance absorption, you could give a lower effective dose, and just the fact that there's less iron unabsorbed in the GI tract should reduce GI side effects.
So we do have 2 more questions, but do want to also give time to the other speakers. So Namajit and who's other Gautam, if you can put those chat in the your question in the chat, I can send it over to Jimmy at the end and get those questions out to you.
All right, thank you.
So now I have the opportunity to announce our next speaker. Wiramon Rungratanawanich is a research fellow in the section of Molecular Pharmacology and Toxicology in the National Institute on Alcohol Abuse and Alcoholism at the NIH. She began there as a postdoc and previously was a lecturer in the Faculty of Pharmaceutical Sciences at Burapha University. Her research centers on alcohol and intoxicant-mediated oxidative stress, post-translational protein modifications, inflammation, and the crosstalk between gut-liver-brain axis. Her career and productivity are reflected in her extensive research output, which includes 26 peer-reviewed papers, a book chapter, and 2 editorials, and 15 oral and 10 poster presentations showcasing her work at notable conferences. Her accomplishments have been recognized in 15 awards that include the NIAAA Daniel Homer Fellow Award for Outstanding Fellow and the Research Society on Alcohol Junior Investigator Award and the NIH Fellow Award for Research Excellence. She received her bachelor's in pharmaceutical sciences from Chulalongkorn, Bangkok, Thailand, and her PhD in neuroscience from University of Brescia, Italy. And so now I have the pleasure of welcoming, and you can share your screen. We're only seeing a portion of the screen. Mhm.
Notice still.
Hey, Michael, do you
How
have,
about it? Okay, does it
oh,
work?
yes, there you
Okay.
go. Perfect. Okay.
Okay, so I start. So, good morning, everyone, and thank you for your introduction. And first of all, I would like to thank you very much for the ODS for your kind providing me the ODS Scholar Award to present my research today. And my research today, I will present only the work that I received the ODS Scholar Award about the overall projects, the results, conclusion, and all of this already published in Journal of Pioneer Plant. So, um, my research title is Melatonin Prevents Gut Leakiness and Liver Fibrosis through the Gut-Liver Axis. My name's, I cannot change. Okay. I cannot change the, okay, change now. Okay. Technical. Sorry for the technical issue. So my name is Wiramon Rungratanawanich, as already mentioned. So it's sound like a we, it's like you and me, and Wiramon. Currently, I'm a research fellow in National Institute on Alcohol Abuse and Alcoholism, NIAAA, NIH. A little bit background about myself. I was born in Thailand, in Bangkok. It's the land of smile. And as mentioned before in the introduction, I received my bachelor degree from pharmaceutical science from Chulalongkorn University in Bangkok. Before I received a PhD scholarship in neuroscience from University of Brescia in Italy, where I had an opportunity to study proteomics analysis in University of Bonn in Germany as well. And then I was selected as a postdoctoral fellow to come here in the United States to the NIAAA, NIH. And it's my honor here to present my research on melatonin prevents gut leakiness and liver fibrosis through the gut-liver axis. So when we talk about liver fibrosis, we know that liver fibrosis is a consequence of several chronic liver diseases that once developed, it is either reversible or results in cirrhosis, and without liver transplant, it can progress to the liver cancer, liver failure, and mortality. In patients with liver fibrosis and cirrhosis, they commonly express a high levels of endotoxin level, which is a marker of gut leakiness. However, the causal role of gut leakiness and its mechanism underlying liver fibrosis, as well as therapeutic intervention through the gut-liver axis, remain unclear. And melatonin. Melatonin is a hormone produced in the pineal gland and is largely generated, you can see around 400 times more in the GI tract or in gastrointestinal tract. Melatonin can regulate circadian rhythms and sleep that result in improved insomnia. It can also act as a key regulator of anti-inflammatory and antioxidant system. Melatonin can scavenge the reactive oxygen-nitrogen species and lactic peroxides. It can increase endogenous antioxidant enzymes such as superoxide dismutase, glutathione, catalase. It can also reduce pro-inflammatory and pro-fibrogenic markers such as CNF beta, NF-kappa B, interleukin-1 beta, interleukin-6, and TGF-beta. Moreover, melatonin can regulate diverse physiological functions such as immune response and tissue organ injury. And melatonin consumption as a dietary supplement can increase serum melatonin and distribute it to all the body compartments, as melatonin can pass through the cell membranes and other barriers. Therefore, melatonin supplementation can not only improve insomnia, but help prevent or protect against injury in many organs. However, the mechanism by which melatonin exerts its beneficial effects on preventing gut leakiness and liver fibrosis remain elusive. Here in this study, we aim to investigate the contributing role of gut leakiness in liver fibrosis through the gut-liver axis and the protective mechanisms of melatonin. So in this study, I used rats as a model, and I treated rats orally like oral gavage with melatonin followed by TAA injection. TAA is thiocetamide. It is a model that used widely in rodents, rats, and mice for decades to induce liver fibrosis. And after tissue were collected and processed for the analysis. And next, the first results I'm showing you is from the gut. You can see here the pink color, this H&E staining to show you the gut structure. The right is the blue color, and it show you the tunnel staining, and the brown dot is for apoptosis. And the first column on the left is from control, followed by melatonin alone, TAA alone, and melatonin plus TAA, which is similar to the right in the blue color. And the upper panel is the treatment after TAA for 1 week, in the middle after 2 weeks, and at the bottom after 4 weeks. As you can see here, that TAA alone caused gut structural deformation from 1, 2, and 4 weeks, which are protected by melatonin pretreatment. Similarly, on the right, you can see here that TAA increased cellular apoptosis in the gut here from 1, 2, and 4 weeks, which are protected by melatonin pretreatment. And then I measure the gut tight and adherent junction protein. And what are these proteins? These proteins are important in maintaining gut-intestinal barrier function. These proteins include CO1, claudin, octreotide, gamma-catenin, beta-catenin, ecadherin. And the decrease or disruption of these proteins results in increase of intestinal permeability. So I measure all of these levels. And as you can see here, I'm showing you on the left is from 1 week, in the middle from 2 weeks, and on the right from 4 weeks. And the column is in the same order: control, melatonin alone, TAA alone, and melatonin plus TAA. You can see here, TAA significantly decreased this TJAJ from 1, 2, and 4 weeks, which are protected by melatonin pretreatment. And you may notice one marker that is highly increased in TAA after 1, 2, and 4 weeks, which is a marker of apoptosis that's called CLIP-TASPER3, and is protected by melatonin pretreatment. Next, I measure serum endotoxin, which is a marker of gut leakiness. As you can see here, the green color shows you the result from 1 week, the red from 2 weeks, and the blue from 4 weeks, and is in the similar order of control, melatonin, TAA, melatonin plus TAA. You can see that from 1 week, TAA already increased gut leakiness and persists until 2 and 4 weeks. And all of these were protected by melatonin pretreatment. But not only in the gut, I want to know what happened in the liver. You can see here on the right, the liver injury markers increased significantly only after 2 weeks, no change in 1 week. You can see here from 2 and 4 weeks, liver injury markers increased and is decreased by melatonin pretreatment. These results were supported by liver histology. You can see here in the orange color, the red color here shows you the higher degree of liver fibrosis. And on the right, similarly tunnel staining, the brown dot show you the apoptosis. As you can see here, that TAA from 2 weeks markedly increased liver fibrosis, and also 4 weeks, and is protected by melatonin pretreatment. On the right, similarly, liver has a higher levels of apoptosis, which were protected by melatonin pretreatment. And these results were supported by the protein markers of liver fibrosis and apoptosis that is increased by TAA and is decreased by melatonin pretreatment. So after we see these changes that TAA increased gut leakiness and liver fibrosis and is protected by melatonin, we want to know that what could be the underlying mechanism under this. Here, in our lab and others, we have shown the aberrant protein function from post-translational modification that could lead to gut leakness and liver injury. So in this study, I measured the PTM's post-translational modifications in the gut. And here, as you can see on the left, is from nitrated proteins. On the right, phosphorylated proteins. You can see that I found no significant changes of nitrated and phosphorylated protein. But next, I measured the levels of protein acetylation. As you can see here, that TAA significantly increased protein acetylation from 1, 2, and 4 weeks in the gut, and is opposite to this third one is a protein deacetylation that is decreased by TAA from 1, 2, and 4 weeks and improved by melatonin pretreatment. And then next, I want to know that what proteins that are acetylated in the gut. Here on the right, I perform immunoprecipitation of gut intestinal tight junction adherent junction proteins such as CO1 and gamma-catenin. You can see here that TAA markedly increased protein acetylation on these TJAJ proteins and followed by ubiquitination. As we know that ubiquitin can mediate the degradation of this protein or proteolysis. And these effects were decreased by melatonin pretreatment. And not only that, I found that in the gut, if TAA increased gut dysbiosis, but decreased short-chain fatty acids from 1, 2, and 4 weeks and is protected by melatonin pretreatment. And after I see these changes in the gut, I also want to know what happened in the liver. Here, similarly, I measure the levels of post-translational modifications or PTMs in the liver. On the left, you can see this from nitrated proteins. On the right, you can see this from phosphorylated proteins. And I found no significant changes in these nitrated and phosphorylated proteins in the liver. But here, you can see there is a higher levels of protein acetylation from TAA from 1, 2, and 4 weeks, and is decreased by melatonin pretreatment, and is also opposite to the Sirt1 deacetylation that is decreased by TAA, but improved by melatonin pretreatment. So after we see this increase of protein acetylation in both the gut and the liver, we want to know what is the process or enzymes that involved in protein acetylation. Here, I'm showing you the simple process of protein acetylation and deacetylation. Protein acetylation involved enzymes that call histone or lysine acetyltransferase that use acetyl-CoA as a substrate. But deacetylation, they are histone or lysine deacetylase or sirtuine or Sirt isoform 1-7 that use NAD as a substrate. So I measure all these markers, and as you can see here, acetyl-CoA, acetyltransferase, histone deacetylase, NAD, and the protein expression. I found no significant changes in these markers. So then I measure the levels of sirtuine 1-7 isoform. And as you can see here, only Sirt1, I showed the result before as well, but only Sirt1 that is decreased by TAA and is improved or prevented by melatonin pretreatment. And not only protein changes, the Sirt1 activity, mRNA expression also decreased by TAA and protected by melatonin pretreatment. And I also want to know that this increased acetylation in the liver, which protein are acetylated. Here, I found the increase of acetylation of Sirt1 targeted protein. These proteins such as NF-κB that is involved in inflammation, P53 that is involved in the apoptosis, and also FOXO1 that is related to metabolism. This protein is highly acetylated by TAA and is decreased by melatonin pretreatment. And next, after I found the changes of increased acetylation and specific decrease of Sirt1. So I want to know and confirm the role of Sirt1 in gut leakiness and liver fibrosis. So next, we perform the experiment using intestinal specific Sirt1 knockout mice, or I will call as a gut Sirt1 knockout or gut knockout mice. And then I inject the mice with TAA. This is a similar model that induced liver fibrosis in the rodents, and the tissue were collected and processed for the analysis. Here, this first result, I'm showing you this from the gut. You can see the upper panel is from control, the lower one from TAA exposure. On the left column is actually from the flocks, but I wrote Y type for the short form. And on the right is one from gut Sirt1 knockout mice. You can see here that gut Sirt1 knockout mice, they have a higher degree of intestinal deformation, even compared to the Y type of flocks that treated with TAA. On the right, you can see that gut Sirt1 knockout mice, they have a lower levels of these TGAJ proteins that is important in maintaining gut intestinal barrier function. It's lower than even flocks plus TAA. And then I measure the levels of serum endotoxin here and serum FITCD4. This is the marker that we gavage to the rodents and to evaluate intestinal permeability. The green color is from flock mice, the left control, the right TAA. The red color from gut Sirt1 knockout mice, the left control, the right TAA. You can see that gut Sirt1 knockout mice, they have a higher levels of gut leakiness even compared to the flocks that treated with TAA in the similar doses. And then not only in the gut, I want to know what happened in the liver. But as you can see, even the gut that the third one that is knocked out in the gut can affect the liver. You can see that liver injury markers, ALT and AST, gut Sirt1 knockout mice, they have a higher levels of liver injury after TAA exposure, even compared to the flock that's treated with TAA. Besides this histology, it is the same PCORS staining to show you the liver fibrosis. You can see that gut Sirt1 knockout mice, they have a higher degree of liver fibrosis when compared to the Y type of flocks that plus TAA. On the right, I confirm you the levels of proteins of the marker fibrosis and apoptosis that is highly increased in gut Sirt1 knockout mice even compared to the flocks plus TAA. And besides, after we see the changes in the gut leakiness, liver fibrosis, I want to know how about acetylation. Here, on the left, I'm showing you this from the gut enterocytes. On the right, this from liver protein. You can see here the Sirt1 level is decreased by TAA, but no Sirt1 in the gut. And in the liver, TAA also decreased Sirt1 expression. But you can see that both gut and liver, they have a higher levels of protein acetylation even compared to the flocks plus TAA. And then I also found a similar results of the increased acetylation of Sirt1 targeted proteins in gut Sirt1 knockout mice, as you can see here. That is highly increased, especially in gut Sirt1 plus TAA on the proteins like NF-kappa B, P53, and FOXO1. And besides these in vivo results, I also have in vitro study. In vitro study, the first one I'm showing you this from hepatic AML 12 cells. I found a decreased Sirt1 but increased protein acetylation in hepatocytes. Just in brief that the green signal on the left is from Sirt1 levels, and the green signal is on the right is from protein acetylation. And the column on the left is from control, TAA alone, and this one is a Sirt1 activator plus TAA, and this one Sirt1 is a Sirt1 inhibitor plus TAA, similar to the right. You can see that Sirt1 activator can prevent the effect of TAA decreased Sirt1 levels, and Sirt1 inhibitor exacerbated the levels of the changes of TAA that decrease Sirt1 levels. Similarly, on the right, you can see that Sirt1 activator can prevent TAA increased acetylation, and Sirt1 active inhibitor exacerbate TAA increased protein acetylation. So after I see these changes in the hepatocytes, I want to know what happened in the gut or intestinal cells. Here, I use human intestinal T84 cells, and all the green signal here, these are from Sirt1, but I have 8 treatment. So the column on the left is from control, and the next one is TAA alone. This one, Sirt1 activator plus TAA, and followed by Sirt1 inhibitor plus TAA. On the right, melatonin alone, melatonin plus TAA, and Sirt1 activator plus melatonin plus TAA, and the last one, Sirt1 inhibitor plus melatonin plus TAA. As you can see here, TAA alone decreased Sirt1 levels in the intestinal cells, which is protected by melatonin pretreatment that treated with TAA. This is also similar to the effect of Sirt1 activator plus TAA that can prevent the decrease of Sirt1 levels. And also, it's even better with the Sirt1 activator plus melatonin plus TAA. But this effect were worsened by Sirt1 inhibitor even in the presence of melatonin plus TAA. This could indicate that melatonin exerts its beneficial effects at least partly through Sirt1 pathway. And next, I also performed the experiment measure the levels of this claudin 1, which is a tight junction protein, to see the tight junction formation. And it is a similar treatment of 8 columns. And as you can see here, TA alone caused the intestinal tight junction disformation or interruption, which is protected by melatonin pretreatment plus TAA, which is similar to the Sirt1 activator plus TAA, and is even better with Sirt1 activator plus melatonin plus TAA. And these effects were exacerbated by Sirt1 inhibitor, even plus melatonin, but plus TAA. And similarly, this adherent junction proteins is similar to claudin 1 tight junction, that TAA decrease intestinal formation of these adherent junction proteins is protected by melatonin, similar to Sirt1 activator and better with Sirt1 activator plus melatonin, and is worsened by Sirt1 inhibitor plus TAA and even plus melatonin. And these results were confirmed by immunoblood analysis, showing you here that TAA markedly significantly decreased Sirt1 levels and increased protein acetylation in the intestinal cells and is protected by melatonin pretreatment. On the right, these TJAJ tight junction adherent junction proteins, we can see that TAA significantly decreased these TJA proteins and is protected by melatonin pretreatment. So from overall, I would like to summarize that melatonin prevents TAA-induced gut leakiness and liver fibrosis through the gut-liver axis via modulating Sirt1-related deacetylation of the gut tight junction and adherent junction complex and hepatic proteins. And lastly, I would like to thank you very much, my supervisor, Dr. BJ Song of our section of Molecular Pharmacology and Toxicology. And thank you so much for your attention today. This is my contact email. Thank you so much. Thank you.
Thank you. We have some time for some questions. You can put them in the chat or raise your hand. Also, I will be putting together a list to send to our speakers, and I'm going to do block with last week's and this and give them. So I have a question of, I was curious if you have tested whether the protective effects of melatonin on the gut barrier are preserved in the clock gene knockout mice.
That's a great question, actually, and I have got this question before. I actually interested in this, but unfortunately, we haven't tested any gene related to clock or circadian rhythm in this study.
What are your next steps for your research?
Oh, that is really great question. Because now we found the protective effect of melatonin in the gut and liver fibrosis. Actually, since we are in the alcohol research, we are also interested in the effect of alcohol in the gut and the liver, and we would also like to know the effect of melatonin. And since I am personally interested in the organ crosstalk. So it's so possible that not only in the gut and the liver, I also would like to extend to test the effects of melatonin in different organs, especially like in the brain, because I also studied in neuroscience before. But that's a great question.
Well, this is more of a personal question. Have you thought about skeletal muscle at all?
Oh,
That
that
was
is very
something that I
interesting.
have,
Yeah,
thought about in the past.
everybody is related to metabolism, and
Yes.
and it is very interesting. I actually have started looking at the connection of the skeletal muscles and the other organ. That is very, very great suggestion.
It's Sirt3, I think that works a lot in the skeletal muscle, but yeah, we could talk offline about that. I just
That would
wanted
be great. That would be great.
ask about that. So how do you think melatonin alters Sirt1? What about like the mechanism of that?
Oh, that is a really great question. I never thought about that, but what I do know is we use liver fibrosis as a model, and we know that in liver fibrosis, even in patients with liver fibrosis, they have the levels of melatonin almost half of the normals of normal levels, and the absorption of melatonin is also poor in liver fibrosis people. So I know that the dietary supplement of melatonin could help in liver fibrosis and some other disease, such as in multiple sclerosis and also COVID-19, and how melatonin alert Sirt1. It is very interesting, but maybe I should look into that more, like how the changes of melatonin can affect the Sirt1 protein level, mRNA level, and also activity. That's really great one.
So I think we had for 1, 2, any thoughts of NAD plus supplements?
Oh, I have found some papers related to that of NAD supplement. That would be interesting. There are many studies of NAD supplement in the effects of and the beneficial effects directly of NAD without melatonin as well in the gut and the liver, that it showed that NAD can protect the effects of the alcohol and also non-alcoholic induced liver injury and liver fibrosis as well. And plan to test melatonin in humans for impact on the same method. Oh, that's very interesting. If possible, I really want to perform the clinical study, but in our lab, we only work with animals. But if any audience have any ability, I would like to collaborate. We are very welcome. Thank you.
Okay, thank you both for presenting to us today. That were very exciting. If I have any more extra questions, I'll send them along to you all. And again, thank you very much. That was great.
Thanks for having us.
Thank you so much. Have a great day.